Hi everyone, this is a pre-recording of a talk that I'll be giving at RECOMB-Seq 2020. Alright, my name is Lucy Williams, and this is joint work with my advisor, Brendan Moomey. The basic idea of this talk is to explain how we are able to update an existing problem, finding maximal perfect haplotype blocks, to a setting where some data is missing or unknown. The data we're looking at is a large set of SNP calls for different individuals from a population, and we'd like to find conserved blocks from this data. It's already known that this can be done in optimal time when all of the SNPs have been successfully called. Today, I'll tell you about how we can approach the problem when the data has missing values. I'll start off with a brief explanation of why we want to be able to do this efficiently. As we've gained the ability to cheaply sequence entire genomes, we have the opportunity to investigate questions of population genetics at more and more granular scales. Previously, we might have asked whether one specific allele is being selected for. Today, we would like to be able to measure selective pressure across the entire genome, and we'd like to do so efficiently and using as much population data as possible, such as in the figure currently shown on the screen. From left to right, we see every base position of human chromosome 2, which contains 242 million base pairs. The height of the bars shows the selection coefficient at each position, giving a measure of the selective pressure there. With data like this, we can see areas of the human genome that are under recent selective pressure at a very granular level. For other species, such as viruses, we could use this information to aid in drug development. Areas of a virus genome that are under selective pressure would be poor choices to target with a vaccine or other drugs, since they're likely to change in the near future. In 2018, a group of researchers proposed a simple way to compute the selection coefficients across the entire genome. Let's say we're interested in the selection coefficient at base position 121 million. At this location, and the regions around it, there is some specific DNA that dominates in the population, but there may be slight variations that exist among certain subsets of the population. We'll be focusing on variants based on SNPs, single nucleotide polymorphisms, or single base differences, rather than other kinds of genetic variation. These genetic variants may span overlapping sections of the genome. For each of them, we can compute the selection coefficient using a model proposed by Chen et al. Then, we simply set the selection coefficient of our position to the maximum selection coefficient of all variants covering it. If we know what these variants are, we can estimate the selection coefficient for each one by evaluating a few equations, taking only constant time. So, the potential bottleneck is computing all of the variants, and that's what the rest of the talk will focus on. In 2018, Kanha et al. Proposed to use a pattern called a maximal perfect haplotype block for these variants, and gave the first algorithm to find all of these from a population dataset. In general, a haplotype is a set of DNA variations that tend to be inherited together. We'll see that these blocks give a precise way of defining such a set, along with a list of all the individuals in the population who had the specific set of variations. The input data is a set of sequenced genomes for k individuals with n SNP calls each. Generally, we think of a 0 as no change from the reference and a 1 as a change from the reference, but in general, the labels could be arbitrary. We can represent this data as a k by n binary matrix. In this small example, we would see that k is 4 and n is 6. A maximal perfect haplotype block is a subset of the input sequences and an interval of SNPs that is completely conserved among the subset and cannot be extended left, right, or by adding any other sequences. Kanha et al. Formalized this definition using 4 conditions. The first condition says that all sequences in the block must be equal, and the next 3 ensure that the block is maximal. Additionally, they define notation to indicate a block as a list of sequence indices, then the starting column, and then the ending column. Let's take a look at some examples to make sure this is clear. First, do sequences 1 and 4 from columns 1 through 5 form a block? We can see that they satisfy the equality condition. They can't be extended left, since they start at the first column. They can't be extended right without breaking the equality condition, and we can't add any rows to the set and keep them all equal. So yes, this is a block. What about sequences 1 and 2 at column 4 only? There's no problem with including a single SNP as a maximal haplotype block in general. This one satisfies equality and left and right maximality, but we could add sequence 4, so it's not a block. Now that we've defined what a maximal perfect haplotype block is, what we really care about is how quickly we can find all of them in the entire dataset. When Cunha et al. First defined the problem, they gave a big O of kn squared algorithm to solve it. But by 2019, Alenko et al. Gave an optimal solution, finding all blocks in the size of the input matrix, big O of kn. They actually gave two solutions, the first using a suffix tree, and the second using a different data structure called a positional Burrows-Wheeler transform, to process the data column by column, using only big O of k time for each of the n columns. We'll return to this method after defining the wildcard haplotype block problem and highlight how the addition of wildcards causes problems with this approach. We can now define maximal perfect wildcard haplotype blocks. We use a star to indicate wildcard SNPs, which we think of as being able to match to either 0 or 1. Again, we have k sequences of length n as input, but you'll note that we've changed the equality condition to the consensus condition. We assume that each column of a block must contain a non-wildcard value. For example, in the dataset here, the block shown contains at least one 0 in column 1. It actually contains 3 zeros, and at least one 1 in column 2. We say that 0, 1 is the consensus pattern for this block, because all sequences in the block can be made to agree with 0, 1 by setting the wildcards appropriately. For example, if we take a look at sequence 1, there's a 0 in the first column and a wildcard in the second column, so this column matches to the sequence 0, 1. On the other hand, a block containing sequences 1 and 2 at columns 2 and 3 does not satisfy the consensus condition because it does not contain a non-wildcard value in all of its columns. In fact, it doesn't contain a non-wildcard in any of its columns. But if we add sequence 3, it would be a block because now it satisfies the consensus condition, and it can't be extended left, right, or by adding any other rows. Without wildcards, there could only be big O of kn blocks. What about when we add wildcards? We use the symbol t to mean the number of blocks in a given input. We can give one bound on t by counting up, for every column, 1 through n, the possible number of blocks that could end at this column. The proof of this bound comes out of the data structure we'll discuss next. We do note that this bound does not appear tight. Our algorithm, which uses a trie-data structure, is output sensitive in the number of blocks that do exist, taking big O of kn time. A trie, so named because it can be used to efficiently retrieve the strings it is built over, is a tree data structure where edges or nodes are labeled with letters, and paths from the root to the leaf nodes are words from the input dataset. In this case, the words are the SNP call sequences. Our algorithm, just like the algorithms proposed by Cunha et al. And Alenko et al., uses the following key observation. At each column, left maximal blocks correspond to internal branching nodes of the trie, built over the reverses of the sequences up to that column. And so, to find all blocks in the input matrix, we can iterate through each column, build a trie using the reverses of the sequences, and find internal nodes. Alenko et al. Achieved an optimal runtime using this method by showing that we don't need to explicitly compute the entire trie, but we can instead store and update a positional Burrows-Wheeler transform, which is just a few length k arrays, and find the internal branching nodes from there. Let's take a closer look at these tries. At the second column, we've built up our trie by adding reverses of rows, such as 0, 1 for the first sequence and 1, 1 for the second sequence. Since we've got all 4 possible length 2 sequences, we've got a full binary tree. At the third column, we have no more repeated sequences, so we have a unique leaf for every sequence in the trie. Again, we can show that we added the path 1, 0, 1 for the first sequence, and the path 0, 1, 1 for the second sequence. Now, let's take a look at how we can use the trie to check for left maximality. If we keep thinking about following these sequences backwards, a branching node means that there were at least 2 sequences that were following the same path, but then diverged. Consider sequences 2 and 4. Starting from the third column, they both have value 0. But when we move to the second column, S2 has value 1 and S4 has value 0, which results in a branch in the trie, indicating that the block consisting of sequences 2 and 4, starting at column 3, is left maximal. Of course, we then need to check for right maximality, which would fail for these sequences because they both have 0 in the fourth column. Now that we've seen the non-wildcard case, let's turn our attention to what happens when we've got unknown values. We might hope that we can directly extend the trie approach by adding all possible consensus patterns for each reverse sequence. Let's take a look at a simple example. Here, the reverse of sequence 1 is star 0. So we'd add both 0, 0 and 1, 0 to the trie. And the reverse of sequence 2 is star 1. So we'd add 0, 1 and 1, 1. However, our definition of wildcard block requires that each column contain a sampled value. So sequences 1 and 2 at column 2 do not form a block. But the trie has 2 internal branching nodes, which would mean that there are 2 left maximal blocks at column 2. In order to account for this, we define a new trie data structure, which we call the extended block trie at column L. We don't explicitly construct this trie, but instead implicitly explore it using a depth-first search, recording the blocks that it produces as we go. The key here is that, rather than adding all consensus paths, we only add consensus paths that can be blocks, plus a few more that are not reported. To do this, we need to keep track of which rows match to each node, and do some checks as we search the trie. To demonstrate how this works, let's take a look at an example input with wildcards, with 6 rows and 6 columns. Suppose we're currently processing the 4th column. In order to avoid adding false positive nodes, we need to keep track of which rows match to the path to each node. So, we label each node with its matching rows. We also label each node with the path that each node represents. Remember that the trie is built over reverses of sequences up to the current column. So the path 0, 1 would represent a 0 in the 4th column and a 1 in the 3rd column. To keep this picture from being too overwhelming, we have not included node labels for the bottom 2 levels of the tree, but they would also have labels. let's take a closer look. I'll go column by column, but note that our algorithm would actually search depth first. The root node has an empty path, and trivially all rows match to it. When we think about adding our first column, which would be column 4, we add both children. Then we check which rows match to each one. For the 0 child, rows 1, 2, 3, and 4 match. For the child with path 1, only rows 1 and 5 match. Looking at the next column, column 3, we add both children of the 0 node. Path 0, 0 matches to rows 2, 3, and 4, and path 0, 1 matches to rows 1, 3, 4, and 6. You're probably noticing that we do have a 1, 1 path in the dataset, and it would be supported by both rows 1 and 5, but it's not in the try. This is because we check for right maximality of its parent, path 1, before exploring the children. And if we go look in the dataset, the block 1 is not right maximal because we could extend it right without breaking equality. At the second column, we would again add the children of our bottommost nodes, labeling them appropriately, and finally we would add the last column. As I said before, or actually the first column, as I said before, instead of containing all consensus patterns in the dataset, which would generate false positive blocks, this try contains blocks only, plus some additional sequences. For example, we saw that the node for path 1 is not right maximal, but it is included in the try. However, we would not output it as a block. In the end, all internal nodes and some leaf nodes are true blocks. One thing to observe is that as we move from parent to child, the child's row label is always a proper subset of the parents. For example, node 0, 0 has rows 2, 3, and 4, which is a proper subset of its parent, path 0. Because each block must be supported by at least 2 rows, and we must lose at least 1 row each time we move down the try, we know that the maximal depth of the try is k minus 2, which helps us derive the bound we saw earlier. In order to implicitly search the try, we use depth first search, only making a recursive call to explore children if they could lead to a block. We can show that it can only take big O of kn time between blocks, thus the overall runtime is big O of kn times t. We tested our algorithm to find all blocks using a human chromosome 22 dataset, with SNP calls for 5,008 individuals. The data comes from the 1000 Genomes project and was also used in the Elenco and Cunha work. Our dataset did not include missing values, so we randomly assigned some SNP calls to be wildcards. As a baseline, we ran the experiment with no wildcards, 0%. In another, we set 5% of all data values to be wildcards, and in the final experiment, we set 10% to be wildcards. We also varied a minimum area parameter, where the area of the block is defined to be the number of sequences in the block times the number of SNPs in the block. The rightmost 3 columns in the table are all averages per block found. While the total number of blocks increases dramatically as we increase the number of wildcards that are present in the dataset, increasing from 100 million to 500 million to over a billion, the total number above the 500,000 or 2 million area threshold grow more slowly. And if we take a closer look, at scatter plots showing the number of sequences per block versus the number of SNPs per block for block area at least 500,000, we see that the general distribution stays the same even as we increase the number of wildcards from 0% to 5% to 10%. I'll end with a few takeaways that I want you to remember from this talk. First, we saw how maximal perfect wildcard haplotype blocks can be used to identify genetic variants in a population in order to compute selection pressure along a genome. Then, we gave the first algorithm to find all wildcard blocks. It runs in big O of kn times t time, where t is the number of blocks in the input data. We also saw experimentally that for larger blocks, the distribution of blocks remains mostly the same as the number of wildcard SNP as we increase the number of wildcard SNPs, suggesting that having unknown SNP values may be workable even when we are interested in computing selection coefficients. We also suggest some open questions. Can we find better bounds on t? What about a faster algorithm to compute all the blocks? Finally, this is ongoing work. We've proposed a pangenomic formulation of this problem, and we're currently exploring an application to a COVID-19 pangenome. That's the end, and thank you for listening.